Hello, everyone, and thank you for joining us. My name is Beth Fry, and I'll be a product manager here at LGC Biosearch Technologies, and I'll be your moderator for today's event, Plant DNA Purification for NGS Applications, an Underestimated Challenge. And before I introduce today's speakers, I just have a few housekeeping notes for you. First, please note that all participants will be muted to prevent background noise during the presentation. We will have a Q&A session following the presentation, and you can enter your question at any time during the presentation in the questions window. And we'll answer as many as possible during the Q&A session at the end of the event. Any questions that we don't have time to answer at the end of the event, we'll answer by email following the event. Also, we will have poll questions during our event today. We hope that you will be able to participate. We think they're kind of a fun way for the presentation to be a little more interactive for everyone. And we will have an email with a link to the recorded event. Keep an eye on your inbox. You will be receiving that following the event as well. That's all for housekeeping notes. So today we can go ahead and introduce our speakers, Drs. Dietrich Köster and Rob Brazos. Dietrich is a global product manager with LGC. He joins us today from Germany, where he is based. And prior to LGC, he spent time at Bioticon Diagnostics and at the Institute for Product Quality in Berlin. Dietrich completed his PhD at the Max Planck Institute for Molecular Plant Physiology at the University of Potsdam-Golm, and he has over 10 years of experience with nucleic acid purification technologies at LGC, where he is product manager for DNA and RNA extraction and purification product portfolios. Rob Brazos is a senior product manager at LGC and joins us today from LGC's Madison, Wisconsin location, where he is based. Rob's experience includes product manager experience at Roche Nimblegen and at Promega Corporation, as well as over 20 years of bench experience in molecular biology, where he held R&D positions working on vaccine development and transfection reagent development. He received his PhD from the University of Utah, followed by a postdoc at UCSF. And Rob has been with LGC for the past 5 years, where he is responsible for managing our NGS products, including library prep and enzymes. And we're very excited to have both of these speakers present today's topic, plant DNA purification for NGS applications. But before we give the microphone over to Dietrich to start us off, we actually have our first poll question. We'll be talking today about lots of different sample types and sample preparation, but we'd like to know what kind of sample types you are working with. So our first question for you is here. What purification method are you currently using for your samples? This could be beads, this could be spin columns, it could be quick lysis, or it could be other. And we'd appreciate if you can vote using your poll question panel, and we'd love to hear what you are working with. All right, and it looks like our audience is using beads and spin columns primarily, around 36% and 34%, quick lysis about 14%, and then other methods 16%. And those of you who are using other methods, if you wouldn't mind, please let us know what those methods are in the questions box, so we can get a feel for what our audience is using currently. And with that, thank you for participating in our first poll. We appreciate it, and I'll hand it over to Dietrich, who is our first presenter for today.
Yeah, hello everybody. Welcome from Germany. In general, when we're talking about DNA purification, I mean, this is really a step in the middle of a process. On the left, always we have a sample. In the middle, there's the purification and extraction process. And on the right, we have usually some kind of downstream application, right? So in terms of a standard ag bio workflow, so a plant breeding workflow, we are often talking about leaf and seed samples, but we also had projects in the past where we are looking for root samples and flower samples. And as our poll has shown already, you know, there are different kind of extraction methods. There might be mag beads, which are quite common for automated DNA purification workflows. But there are also more and more coming quick and easy methods like quick extract, one of our products which we are using here. But also there's precipitation and there are standard spin columns, right? And on the other side, we have the downstream application, right? So really, we're talking on focusing on next generation sequencing applications today. But there's for sure also PCR, but the bioships, microarrays. So again, whole message here is DNA purification is really in the middle of the whole thing. And I really would recommend always that there need to be starting key considerations. That means I think there are 5 main points where you really should take a step back and think about them. What do we have here and what are our goals here? And I will come to the starting material, which is quite important for a successful NGS workflow, because this is really, I think, one of the key things that you think about your starting material, because if you're not having a good sample type or a good sampling process at the beginning, you cannot expect good high quality DNA, which then needed for your NGS applications afterwards. That's the reason why we have this webinar today. But there are also other goals and objectives. What are the backgrounds? What kind of strategies do you have for your desired outcome? Pricing is always super important, because for sure we all know that plant breeders in general are quite price sensitive. You have maybe tight budgets, not a lot of money, and we simply are often working together with plant breeders right from the beginning to understand their pricing needs. And then often we can simply do specific offers or customized offers which are fulfilling your budgets, right? So we are always saying, when we're starting together a project, we are quite open about prices right from the beginning. The throughput is important because plant breeders in particular have also often needs of a breeding peak. That means that you have a lot of samples in a short time period, and often that means that you need some automation here, right? And the automation then is adding another factor into this whole workflow thing. And finally, the downstream requirements. Again, today we are more or less focusing only on next generation sequencing, but might be other needs. For example, NGS, think about PacBio instruments or Oxford Nanopore applications, where you simply have really needs for high molecular weight DNA and not just short fragments like Illumina applications, for example. So again, this is really a complex scenario, and that's the reason why we have called these kind of webinar today Underestimated Challenge. Again, I really would like to underline that here at the beginning, that we are really looking really, and we are really recommending to have a look on your sample type at the beginning, simply because plant cells really make the DNA purification step sometimes really challenging. Simply the age of the sample is quite important. Younger leaves are often easier to lyse than older leaves, but also in terms of seeds, there's a lot of variation there. It could be the size of the seed. Just think about, for example, a canola seed and a sunflower seed, right? The size is really makes a difference here, and it is depending, and this is then really having a high impact on your grinding conditions. So what kind of steel ball do you use? Maybe a 3 millimeter or 5 millimeter steel ball. But also starch and oil is often important, right? So there are simply oil-rich seeds like canola or sunflower seeds, and then the extraction process also needs to remove these fatty acids and these oil layers in the vials very efficiently. Otherwise, you might run into issues. So there's really also seed variation. Leaf variation, dried and fresh, old versus young, there is really a lot ongoing there. We also had rotten leaves once in our lab. That was a very special project. It really depends how your sampling process looks like. You know, sometimes you can just go from the field to the lab, because the field is quite in the neighborhood of your lab, but sometimes you have to ship the leaves maybe from another country to a core facility and so on. And that makes an impact, because if you need to ship these samples, you need to dry them. If you do not need to dry them, then then you can take fresh leaves maybe, and then, for example, you can play around with the lysis conditions a little bit, using less lysis buffer, for example. And finally, the sample quantity impacts the purification. That means, you know, we all know NGS might require higher DNA yields, and therefore you might need to start with more starting weight. That means maybe a couple of leaf punches, but sometimes even one leaf punch is sufficient. That depends again on your requirements downstream of the downstream application, but also depends on the sample. For example, we know that young leaf samples often contain more DNA because these samples are photosynthetically active, in contrast to older leaves who are simply not as active as fresh leaves or young leaf samples. So again, all these kind of things have an impact here, and it's super important at the beginning that you simply are aware of that.
Okay, and that will bring us to our second of our 3 poll questions today. First, we asked about what kind of purification methods you're currently working with, and now we'd like to know what kind of sample types you're currently working with. Please use the voting buttons to select one. Maybe you're working with grain crops like corn or wheat. Perhaps you're working with vegetable crops like tomato and pepper. or maybe it's horticultural species like trees and flowers. Maybe you're working on fruits, or maybe you're working on other things like palm oil. Please let us know what you're working with. And again, if you have something that is not listed here, we'd love to know what it is. Please let us know in the questions box. We'd be glad to know what kind of sample types you're working with. All right, looks like the majority of our audience today is working with grain crops. About 43% of our the folks that answered, Dietrich, are working with grain crops, followed by a pretty even mix between vegetable crops and horticultural species. And then a pretty good portion, actually more folks that are working with things that we haven't listed here, others. So yeah, please do let us know what some of those folks, what some of you are working with in the questions box. Thank you.
What is super important is the downstream application as well, right? I've spoken a little bit about the sample already, but now let's talk about the downstream application. Here you simply has to distinguish between next generation sequencing and PCR, because simply the priorities are totally different, right? For NGS, it is really a must that you need high quality DNA or high quality nucleic acids, DNA and RNA. And therefore, really, this is super important that the quality is correct. And if you compare this to PCR, you know, when they have this hundreds of thousands of samples per year, simply it is simply a bigger thing in terms of throughput, right? Because you have so many samples, you have a breeding peak, and you simply would like to screen these kind of plant populations as soon as possible, and therefore throughput is simply super important. And throughput automatically means automation, right? You need simply maybe a medium to high throughput machine which is helping you to get rid of these samples which are just flooding your lab. But there are other things as well. The throughput for sure is also important for NGS. Why? Because Illumina is steadily launching new instruments all the time. I mean, I've started to work for LGC 10 years ago, and there I think we had just a very one or two machines here in our service lab. But nowadays, you know, there are so many different kind of machines. Illumina is launching constantly new machines, new instruments for different kind of applications, including high throughput instruments. And there's a clear trend as well to use sequencing also as a high throughput tool. And Rob will come back to that maybe in his part. And finally, cost efficiency is super important. For PCR, definitely, because you have so many samples, so that you're looking simply for the most effective way and most cost efficient way to get rid of your samples or to extract your samples, but also for NGS, again, because if you have more samples, unfortunately, probably your budgets are not growing in the same way as your samples do, right? So again, be aware of that. And again, here we are really talking about high quality DNA and RNA. Going a little bit more into detail in terms of sbeadex. So first of all, we are having a proprietary sbeadex workflow. That means that we are following not the standard workflow, lyse, bind, wash, and elute. We're having a second binding step. And why do we have that? Because we are using a slight different binding technology in comparison to our competitors or other suppliers in the market. What we're doing is we are binding the DNA in a first binding step to one first silica coating, which you can see here. So we are binding the DNA to this X's. The X's are representing a coating. And then we have a second coating, which can be activated by one of these buffers, which I'm showing you on the right. And then we are changing the binding technology to an adapter. That means we are binding the DNA not anymore to a surface, but we are binding it to an adapter, and the adapter then is binding to the surface, so that we have some kind of bridge here between the DNA and the coating. And that has one main benefit, because you know, often you have a problem with ethanol carryover, because this final wash step usually is based on 70% ethanol. In our case, it's a water-based washing buffer, and therefore you do not need to evaporate your ethanol anymore, and you can go straight after your washing into the elution step. So you have some kind of time benefit here, because you do not wait to dry your DNA, and you have no risk that any ethanol is carried over in your final DNA preparation, right? And that is simply giving you a big boost in terms of DNA quality, and is a big benefit simply because you don't have this risk here, and you don't have any PCR inhibition and so on. Yeah, so as a take-home message here, our beads are double coated, and we are really using no ethanol in the final wash buffer, so that you not have any ethanol carryover. This is just giving you a brief idea what is in the kit. Again, what is just important, we can scale up and scale down the kits according to your needs. Usually we have two different kind of formats, the mini and the maxi format. The mini format is based on the assumption that you're starting with 20 to 30 milligram of sample weight material, so 20 to 30 milligram of leaf material, for example. The maxi kit is for 80 to 100 milligrams, but honestly, we don't care if you're starting with 50 or 40. Again, we are always very flexible, and we can offer you a customized kit if you want, right? So, but it's just to let you know what we have here. Also, in terms of bike sizes, we are usually offering different kind of standard off-the-shelf bike sizes like the 960 kit, but we're also offering bulk formats for 5, 10, and 25,000 purifications per kit, right? So that's up to you. You're totally flexible here. We can simply you can simply choose whatever you need. So let us know, and we're doing it. And also what I want to mention is we have a toolbox, because it's quite important for the results I'm showing you in a minute. We are offering different kind of lysis buffers. We are offering different kind of binding buffers, wash buffers. And this is giving you an idea about our toolbox, how it looks like, right? So what kind of buffers do we have? And in the end, we are testing out which kind of buffers are fitting best for your sample type and for your requirements you need. That's what we do usually, right? But I'll come to this later on. There are also a couple of things which are not included into the kit, like protease, like tips and plastics and so on. But yeah, that's, I think, the standard thing here. And again, what's quite important is, in particular, when you're talking about high throughput DNA purification, is that these kind of beads are running on different kind of platforms. In our case, you can use our sbeadex beads for on all open platforms. Might be a Hamilton liquid handler, might be a Kingfisher, but also we are offering these our high throughput workhorse called the Octopure, which is capable to purify 896 plates in 3 hours, which is then already going into the high throughput area, right? So in the end, it doesn't matter what kind of machine you're using. Maybe you have one already in your lab, maybe you are thinking about buying one, and we can give you also their advices, what machine fits best to your needs today and maybe in the next years. So because we are working with these companies closely together, so if the Octopure, so our instrument, is not a good fit for your needs at the moment because your throughput is maybe too low, then we can also always are very open for other instrumentation. Yeah, so it's not a must that you're using the Octopure, but again, we have customers who have started with one Kingfisher, have then bought a second and third instrument. In the end, they have now an Octopure after 5 years, simply because their throughputs were growing and growing. And for sure, you can use these kits also manually. And finally, from the instrumentation perspective, again, this is giving you a close idea about the Octopure. Again, it's a high throughput instrument. We have engineered it for, simply because there was a clear need for an instrument which can process more than one plate in parallel. In this case, Octo 8 means that you can process 896 plates in parallel, so that approximately after, let's say, 2.5 to 3 hours, you've purified about 750 samples. For sure, the DNA quality should be very good. Should be used for NGS applications. Can be used also PCR. We have placed around 100 systems of this robot in the market so far. It's being used for COVID as well nowadays. That's something why we have been so busy in the last month. But again, so in the end, you have getting a package of automation and speeds. And with that, I would like to hand over to Rob.
All right, thank you, Dietrich. Great intro to what I'm going to talk about today. So hopefully everybody can see my screen. If you can't, let me know. But my goal here is, in this short number of slides, is to really talk about expanding on what Dietrich was mentioning about the importance of high quality DNA and RNA in a library prep, and I'm going to and GS, and I'm going to talk mostly about DNA today, since that's most of the work in ag bio. So what are we talking about here? So what I wanted to do was split it into two basic applications: whole genome sequencing, as you could see here on this slide, and then into targeted sequencing using something like PCR or hybridization-based enrichment. And I'm a picture guy, so I like pictures because it helps me understand what's going on. I hope they're valuable to you as well. But this slide just gives you an overview of sort of a typical Illumina DNA library prep, where we all start with genomic DNA. We've got maybe 3 different regions here in different colors. That DNA is fragmented either by sonication or mechanical methods or enzymatic methods, okay? And what you end up with is a series of small fragments, because that's what works on Illumina instruments. And they don't have perfect ends. So they're going to have overhangs. Some will be blunt, 5' overhangs, 3' overhangs. So the first step in library prep is usually to end repair and A-tail those fragments. So the end repair makes them blunt. There's also a phosphorylation event that's there to make sure the 5' ends are phosphorylated that I'm not showing. And then finally, you add an A-tail, and you add the A-tail, 3' A-tail, because the aluminum adapters are almost always 3' T-tailed. Okay, so you leverage that one base of complementarity to drive the ligation of the adapter to these library fragments that you've just end repaired and A-tailed. Okay, and now in this slide, this is the perfect world, right? In this case, what we've got is we've got every single library fragment that I'm showing, or input fragment that I'm showing here, being converted into a sequenceable fragment in the library. And what that means is they've all got the aluminum adapters at both ends, okay? If they don't have the aluminum adapters at both ends, we all know they don't sequence, okay? So they're lost. So in this case, we would call this a 100% efficient library with these, 15 fragments. And that means that all of those fragments are sequenceable, and that's going to give you the best sequencing results, whether you're doing de novo sequencing with whole genome libraries or resequencing looking for new SNPs or variants. Really, this is the crux of everything. So you might even add in a PCR step here, right? But if you don't have those fragments with adapters on both ends, the PCR is not going to help you. So the more complex this library is, in other words, the higher percentage of fragments that are converted into sequenceable material, the better. So that was a perfect world. Now what happens in real life, and because of the things that Dietrich talked about, where you've got poor quality DNA going into your library prep kits because you're not using a sbeadex kit or you're using another kit or method, what happens in real life is shown on this slide. And the bottom line is a lot of things can go wrong in whole genome library prep, okay? You can have problems with ligation, so everything looks good except the ligation doesn't work. You can have poor A-tailing, and so if you don't have an A-tail, your T-tailed adapter is not going to ligate to those ends. And then finally, you could just have straight up poor end repair that didn't happen early on in library prep, okay? And obviously, if it's not a blunt end that's A-tailed, it can't ligate onto the adapters. So in this particular scenario, we've got a 60% efficient library construction. So about 40% of the fragments that were input into this library prep are not sequenceable, again, because they don't have the adapters at both ends. So what does that mean? Well, that means you lose information from your library. You have the potential to lose information from your library. So there are fewer sequenceable fragments, and that information, if it's not in the form of a library fragment, it's lost, right? You can't sequence it. Now there are ways around this. You could sequence, you could use a lot of DNA as input. You could sequence really deeply to find those areas that might have been, converted into library, but very poorly. There's a lot of things that you can do, but ultimately we all want this to be as streamlined and as efficient as possible. So the better the library, the more complex those libraries, the better everything happens downstream, okay? Better multiplexing, better data quality, you name it, better enrichment, you name it, okay? So that's whole genome sequencing library prep, and that's sort of a classic method that we all use. Now, in ag bio, it's very common to do targeted sequencing, and this could be targeted sequencing by PCR style, amplicon-seq. It could be hybridization-based enrichment using probes to pull out regions of interest. We'll focus on sort of a PCR style here. It doesn't really matter. What we're going to talk about applies to everything, okay? So here's my schematic. These 4 regions in the 4 different colors are the target regions. And in a perfect world, you'll do, let's say, in this case, a PCR-based amplicon-seq. And what you get are every target region is represented perfectly. Okay, you've got, in this particular example, this is my very simplistic example, you've got 27 sequencing reads that you're analyzing, okay? So every target region represented by the individual fragments is represented 3 times, okay? So you've got a total of 27 reads for a 3x depth. Really, really good. Perfect scenario again, perfect world. Again, we know that doesn't happen, right? So this next one is just an example of some off-target reads. So you had really good uniformity across your 4 target regions, but you started to see some non-specific amplicons that were produced that take away from the reads across your target regions, okay? So what do you get here? With the same number of sequencing reads per sample, On the instrument, now you're down to 2x depth across your target regions. Ooh, that's bad, right? So that means you won't be able to multiplex as high, or you might have regions that you can't analyze because you don't have enough depth across those regions. If, for instance, 3x was your limit, okay? And then in the third scenario, which is even worse, you have everything going on. So you've got some off-target reads, and then you've got uneven amplicon seek. So these target regions are overrepresented or underrepresented. Okay, so what is that? So what? Why do we care? Okay, well, let's look at that on the next slide. So in this hypothetical experiment, it's done on a very small sequencer when you're looking at 27 reads per sample or so, okay? And we're only pooling 12 libraries in my example here, but I didn't want to draw too many lines, as it turns out. So in this case, we're looking for 3x coverage, okay? And across these 4 target regions, we've got 27 sequencing reads, as we've shown here. Perfect situation again. This is what we talked about before. And in this experiment, because you've got a really tiny sequencer, you can pool 12 libraries to get your 3x depth. Okay, 12 times 27 reads. Very, very small sequencer. All right, then in suboptimal results number 3, where everything was kind of going off, where you had lower coverage, higher coverage, you name it, and a few off-target reads, we're obviously not hitting that 3x depth on all of our target regions. So we are on this one, and it's overrepresented, and this one is spot on at 3x. But we know, and our goal is to get 3x coverage across all the targets. So what do we have to do? Well, we have to sequence 3 times deeper now with this particular example. So we're up to 81 sequencing reads for this sample, this library, in order to get a minimum of 3x reads at that poorest region. Okay, so now you can see everything's sort of proportional. We've got super overrepresentation of this region. We've got overrepresentation of this one, but we're still now at that 3x cutoff, and then this one's overrepresented again. So basically what you're doing is you're wasting sequencing real estate, okay? You're wasting space in the sequencer. And in this particular example, we used to be able to pool 12 libraries in a perfect world, but because of the off-target effects and the uneven representation of the target amplicons in the libraries, we can now only pool 4 libraries to get to our 3x minimum coverage. So it's, it just highlights how important the quality of your targeting amplicons are when you're going into a sequencing reaction, or a sequencing run. So you can improve your multiplexing with better libraries. You can therefore speed up your throughput. You can lower your cost per sample because now you've got more samples on the sequencer in a single run. So everything gets better, the better the quality of those libraries, okay? And one of the key factors, and that's what we're talking about today, is the quality of the DNA and RNA that you're using in your library prep that goes as your input into the library prep. And this slide is just sort of a summary of what might be happening. And again, we're talking about really only DNA today. But most plant samples, as Dietrich pointed out, have inhibitors, right? They have enzymatic inhibitors that are terrible, and every plant sample is different, which is a huge problem. It's not like working with human cells over and over again. They're all different. So purification is key. All right. So what are some of the problems that you could have? So when we're talking about general library prep, where the first steps are adapter, end repair, or adapter ligation, you could have enzyme inhibitors that might inhibit kinases, polymerases, ligases, okay? And what's their impact? Well, they could reduce the complexity of the libraries, okay? Remember, we talked about complexity being super important. If you reduce that complexity, you're gonna ultimately reduce the quality of your results, okay? It could also be that if you have enough inhibition of enzyme activity, you could actually reduce the yields of each library, and that could be problematic in the end, not having enough sample to run on the sequencing run. Okay. In general, if you're doing PCR amplified library prep for whole genome sequencing, it's not that big a deal because there's usually a cleanup step with beads in between. So if you did have some inhibitors here, they're likely not to affect the PCR step. Okay, so we can ignore that. But what are some of the possible solutions? Well, obviously we wouldn't be talking about it today, but if it wasn't one, but the quality of your DNA purification is key, right? The chemistry that you use, the methods that you use, the workflows that you develop, those are all critical to producing the best quality DNA possible. And the better it is, the better your results are going to be. So look to improve that step if this is a problem for you. You could also use a library prep kit that might be resistant to inhibitors, okay, and more robust in general. So even if it's not resistant to these inhibitors, if the efficiency of that adapter ligation is really good, if you have a few inhibitors in there and you drop things down a little bit, you're still going to be okay. All right, so you're still going to have enough library fragments and enough complexity to get the results you want. So it'll be dinged a little bit in terms of the number of fragments created and the complexity, but it won't be so dramatic as if it was a really poor library prep kit. Okay, and obviously we're not talking about our kits today, but we're big fans of our NextSeq kits, and they kind of hit on all these potential problems that you might encounter and might be able to solve with our kits. So definitely look them up if you have a chance, or talk to me later if you want. Okay, amplicon-seq, presence of PCR inhibitors. We all know that they're there. No real impact on whole genome library prep, as I said, because the amplified libraries are cleaned up. But what can happen? You can get low yields of PCR amplicons. You can get uneven target amplification, and related to that, you might just reduce the specificity. Okay, so because some of those molecules that are there, they might have effects on primer binding and/or enzyme activity, which could all lead to specificity issues. So what are some of the solutions? Well, you could improve your purification, again, just like with whole genome library prep. You could look at master mixes or PCR enzymes that are resistant to inhibitors, and they're available. And you could actually upstream, before you start PCR, you could actually clean up the samples with bead cleanup steps like AmpPureXP beads, or with our sbeadex SAB beads, which are unrelated to the beads that Dietrich's talking about, but they're for cleanup and size selection. Those are all opportunities. Now, the one thing that I forgot to put in here for both of these problems is the other thing you could do is you could dilute your samples. This is a classic method with certain sample types that have, like blood samples, blood genomic DNA, that have nasty inhibitors in them. You can dilute them down and use less input material in your enzymatic reaction steps. Now, most of the time, people don't always think more is better. In this case, obviously it's not. So you could back off. Now, what do you need to be able to do that? Again, you need a library prep kit that can handle that lower input amounts. So keep that in mind when you're working with your different library prep kits, okay? And again, the NextSeq kit can handle that. And then finally, what's another problem? We didn't talk about it when I walk through those two workflows, but one of the other problems that you can run into is that your DNA fragments are really small. So whatever you purify from your samples is fragmented, or it started out very small as degraded samples. And what does that do? Well, depending on your kit and depending on your ability to clean up the kits, you might have more adapter dimers and primers present, which can affect the quality of your libraries. Again, if you have those, it all comes back to multiplexing. You're not going to be able to multiplex as much because you've made small library fragments. You're going to need more reads per library to get the same depth of coverage as if you had larger fragments, okay? And the same coverage and depth, okay? So that can also affect PCR-based targeted sequencing, right? If your fragments are so small and your amplicons are too big, you'll start to get dropouts in your amplicons because you are unable to PCR amplify those fragments if the input material, starting material, is too small. So again, that leads to lower multiplexing, poor, less uniform coverage, more dropouts, and so forth. So what can you do? There's a couple things you could do. Obviously, you could utilize a cell lysis usually method that is more gentle, and a DNA purification method that leads to the production of larger fragments, if it was a matter of the kit that you were using and the method that you were using that led to the fragmentation. You could also go back and redesign your targeting primers. So, excuse me, you could decrease the size of the amplicons so that you have a higher likelihood of being able to find fragments that are amplifiable, okay, that you could produce amplicons from. So that's probably, those are probably the two key points there.
Yes, thank you, Rob. So this is our third and final audience poll question for the presentation today, asking about which NGS applications you are using in your next NGS project. And this is a multiple choice answer, so you can choose all of those answers that apply. So perhaps you're planning on using RNA-seq. Perhaps you're planning on using targeted RNA sequencing like hybrid capture or PCR-based targeted RNA sequencing. Maybe you're doing WGS, whole genome sequencing, or whole genome resequencing. Or you could also be using classic GBS, right? Classic genotyping by sequencing. or another type of targeted DNA sequencing. So please let us know which methods you're planning on using. If you're planning on using multiples of these methods, let us know that as well. And if you're using something that isn't listed here, again, please let us know in the questions box. We'd love to know what you have in mind for your next NGS project. All right, and it looks like we have the majority of our audience by a smidge here. We're using classic GBS, genotyping by sequencing, followed by whole genome sequencing or resequencing, and then RNA-seq. Actually, no, I'm wrong. The largest portion of our audience, sorry, is using targeted DNA sequencing. About half of the folks, targeted DNA sequencing. But a nice mix, lots of people using either multiple methods here or a good spread among the different methods that are listed.
I think the last 10 minutes of this presentation day, I would simply would like to give you an idea how a typical application project would look like, right? I've mentioned already that simply a lot of things here are really sample specific, simply because there's such a big variation in terms of sample size, seed, leaves, and so on. And therefore, this sample variation is really key. For example, if you compare a typical DNA extraction pilot study of 2 or 3 different kind of tomato breeders or so on, you probably need 3 different kind of protocols. For sure, you would have one standard protocol, but in the end, the requirements are totally different. Starts with the automation and so on. So again, sample variation is key, and often you cannot really compare maybe, let's say, different kind of seed samples, although they might be the same species. The scientific goals can be totally different. Starting rate I've mentioned already. And I think in general, you know, I think there are 2 general rules. So first of all, who cares about values if the final results look great? Sometimes it's simply not possible to get lovely 260 to 80 values, right? Although you might do everything to get 1.8, 1.9 for the majority of your samples. It's simply not possible, because again, if the sample quality is not good, you will also not get these lovely values. But to be honest, in a lot of cases, who cares about it if the library preparation or the PCRs or the NGS workflow works nice? I mean, then you don't care, right? And you're getting simply experience with these kind of things, and then the values are getting simply less important. The other thing is, always keep in mind, automation can really be a game changer, right? So you can develop and optimize develop the best protocol on earth, but then you still need to automate this, and it makes really an impact if you're using a magnetic particle handler or liquid handler, or it makes an impact if you're using different kind of grinders and shakers and heating devices, and that's really having an impact. So always be aware of that automation is really then changing again the whole scenario, and that's simply important, which makes the whole thing again very complex sometimes. I've mentioned already what kind of toolbox do we have. We are really offering custom tailor-made protocols. So really the majority of our users and researchers who are buying sbeadex are getting their own customized tailor-made protocol, because simply it maximizes the performance with different kind of various samples, right? So I've mentioned already, we have lysis buffers for plants. We have 3 different ones. The difference between these lysis buffers is often maybe different kind of detergents, concentrations, salts. Sometimes you also think about the grinding. Maybe again the steel ball of or the diameter of the steel ball is important, how long you grind, and so on. So they're really, the lysis step honestly is really the most critical ones. If you compare the workload, what our application specialists and researchers in the lab are doing with the rest of the steps, I mean, the lysis step is really probably taking 50% of the optimization. So that's really where they invest a lot of time, because if the lysis is not working well, you can often forget the rest as well. So lysis is really the most important step in this process here. Yes, for sure, you can go also for some different kind of washings, patterns and steps, but I will show you now in the next slides how that looks like. In this case scenario, we have tested 3 different kind of plants. I think 1 grain, 1 fruit, and also some vegetable. And this is how this matrix looks like. Again, it's quite complex here, but I just would like to guide you through what we're doing, right? So first of all, again, we have 3 different kind of plants or sample types, leaves and seeds. And the first thing again is that we're testing different kind of lysis buffers. In this case, it's LPPN and the lysis buffer PVP. So PVP for sure is known as being very efficient against polyphenols and other inhibitors, which are a PCR, which are really a problem, or which might be a problem for the PCR. But usually we have even again 3 lysis buffers, so we would simply add then also another line here with this other lysis buffer we are sometimes using. But on top of that, we have also different kind of binding buffers, right? So it's not just our standard plant lysis buffer. Sometimes we are taking also a lysis buffer from the blood kit, from the sbeadex blood kit, and use it. We can play around with the washing patterns. Yeah, for example, we have 3 different kind of washing buffers: A1, PN, PN2, and then you can start maybe with A1, do a double wash with A1, and go for PN2, or you start with PN1, PN2, PN2, right? So you can see this here in this line, how different these patterns can be, and we are simply testing every combination here with every lysis buffer, every binding buffer. And again, handling automation issues are often decision-making factors, right? So it is maybe the pattern looks nice, maybe the results looking nice on the nano drop afterwards, or maybe your 260 to 80 values look nice, but in the end your beads are clumping, and then you are running into issues with mixing, with shaking, and with the extraction itself, right? So there are also handling and automation issues, which are often a very important factor here. Now, if you have a look here on this table, you see there are some lines which are looking pretty good, and there are others which are not looking good. And for sure, we're not continuing with those who are not looking good, but we are simply taking these lines or these approaches which are looking good and going then to the next level, because we can also play around with other things. For example, we could use debris capture beads, right? Debris capture beads are beads which are pushing down the cell debris, so floating cell debris, which are still in the lysate, so that you're not getting a risk that your tips are blocked with plant residues, for example, cell walls, which are still in the lysate. These are non-magnetic beads, and they're clearing the sample lysate very efficiently. Or we can play around with the lysis volume in general, right? Because a way to increase DNA yields would be not just maybe to really play around with all these washing patterns and really go to the maximum here. The easier way might be simply to increase the starting weight and also increase the lysis volume in general, right? So that you simply get more out of, you get more DNA out at the end. And then again, maybe after washing patterns, you're simply going for the strongest candidate, right? And combining everything here, right? And that is, again, really a typical pilot study. We are spending a lot of time in this kind of optimization. We are offering this also as a collaboration with us. So we have labs in the US and in Germany who are optimizing and testing these kind of sample types together with you to develop the most or the best protocol which fits to your needs. And that's, I think, really something which makes us unique, because LGC or Biosearch Technologies, we are really strongly focusing on this ag bio segment, and we know in all detail what it needs and what your problems are. Finally, and that is now really the worst case scenario, you might have optimized everything and it looks pretty nice, and then you're running your PCR and you still have inhibitions, right? There are simply inhibitions which are not detected by qubit or by the nanodrop. Although your yields are looking nice, the qubit value is looking nice, in the end it doesn't work. And then polyphenols are a good example, and then you might need to go back and change your lysis buffer. For example, you could add extra triglycerol with a reducing agent. Maybe you're adding extra PVP, and that can happen. Honestly, I think in my 10 years with LGC, this just happened, I think, 2 times, right? So that usually you have a very, very good chance that this kind of optimized protocol is also then providing good PCR amplification results. But again, there are unfortunately, some special friends in the ag bio world which are sometimes really challenging. and we know that. We have developed about protocols for about 100 species up to date. So we are pretty experienced, but again, you never know. And as I already mentioned, there are simply new friends all the time, which are sometimes challenging. And then we need to develop this and help you and customers in the optimization. I will come to the end now. So what I would like to say is, you've seen that there is really a lot of work in this optimization. So it can really take some time. And I just would like to underline what is the typical lead time of a project. Again, we are really working very close to our customers, and because we are also experienced in testing new things, often customers are shipping samples to our labs, and we are simply co-developing and co-optimizing protocols together. And these are collaborations which we really like, because it gives us an idea about our chemistry, what we can learn. And it's also making your life easier, because you do not need to do everything by your own. So we are really actively supporting you with the optimization step. Again, the most important first step is really to understand what are your scientific goals. So what are your requirements? We can do this with a questionnaire, with phone calls, with meetings for sure, when you are again allowed to travel after COVID, hopefully very soon. Yeah, so we need to really define what are your needs, what are your requirements. And in the end, what is the outcome of this whole thing? It is a customized protocol. That means it is really a protocol in a chemistry which is fulfilling 100% your requirements. You're also getting a new part number. The configuration is according to your needs. That means in terms of bulk sizes, again, you are really flexible. You can choose whatever you want. We can also add an extra bottle, for example, of lysis buffer, if the optimization study turned out that you need one. So this whole thing is super flexible. And by the way, what is really important for us, that we're only sharing this protocol with you. So you're the only person, or your company is the only company who is allowed to order these kind of kits. And this is really a nice thing. What I would like to underline at this point is, yes, this might be a very nice thing, but again, it might take some time. So please do not expect that this is done within a week or so. If you include the automation aspect on top of that, again, it can take easily 2 to 3 months, simply because the protocol might be not the problem, but again, you need to validate the whole thing on your robot. And then you might have several sample types, or you would like to develop one protocol for all of your crops. So again, this is something which can really take some time. This is my final slide for today. This is the summary. So in the end, again, everything what this is about is high DNA quality. We need high DNA quality so that we have really nice NGS results in the end. And these sbeadex kits, so everything what Rob and I have shown you today is really engineered and developed to get this high quality, and we are really doing everything to support you with that, so you're not alone. I hope that this is really clear to you. Again, and these collaborations are pretty important for us, because again, we are learning as well every day. What is, I think, a unique selling point here is really a USP is our flexibility, right? So we are really the only ones who are offering this kind of the support, but also the scalable protocols, the customization as well. We are offering this tailor-made protocols, and we are simply always open for discussions to sit together and understand what you really need and what you don't need. So you don't need to buy extra bottles. You don't have any leftovers of some buffers. That's simply not the case here. And this flexibility is really the package what we can offer, and which we would love to explore together with you. And so far, I would like to thank you for your attention. I hope this was informative for you. At least we had a great time. I apologize again for the technical difficulties we had at the beginning of this. Now I would like to go back to Beth, and if there are any questions we can answer in the last 5 minutes. Thank you very much.
All right. Great. Thank you, Dietrich, and thank you, Rob, for the presentation. That was fun, and thanks to our audience for sticking with us through the presentation today. And we would like to take your questions. We have some in the questions box that have been asked during the presentation, so I'll shoot these to Dietrich and Rob, and we'll get some answers for you. So I'll try to take them in chronological order here. So we have a a customer who is struggling with alfalfa seed, which is high in starch. Is there anything that you would immediately recommend, and this is in the extraction realm, for samples with high starch or high oil, like palm oil?
Yeah. Palm oil is definitely something. What we're not doing is adding any enzymatic cocktails or so on. It's too expensive, to be honest. It's not necessary, right? I think the palm oil thing is definitely something for the automation as well, because you can simply set up the robot that you're teaching the robot, and you're just not touching the oil thing at all, right? We are having a lot of protocols for oil-rich seeds already for canola. And usually we are just recommending our standard lysis buffer here because it works pretty well also for oil-rich seeds. And we are simply trying to avoid these kind of whatever enzyme cocktails that makes no sense. I think let me check with our application team here what we can do, because without knowing any detail here, for example, how many oil-rich seeds, it's quite difficult to answer that. In terms of the starch-rich thing, it's the same thing here. We have a running protocol for alfalfa seeds. And we are aware that that's not easy. In this case, I think they've added a centrifugation step and with the debris capture beads, which help to pull down the starch granula very efficiently. So in this case, I would strongly recommend really to have a look for these debris capture beads, which I mentioned, the non-magnetic beads, which are really helping up clearing the lysate, and then you're not running into these problems. I think that's what I can say for now. But I think in both cases, I would say let's have a chat afterwards offline.
Okay, sounds good. We can connect you with this customer. Another question, the sbeadex method, does this avoid high molecular weight DNA fragmentation?
It does not avoid it, no, not at all. But you can set up the lysis conditions so well, and the grinding conditions so well, that you can go up to 50 to 100 kilo base pairs as well. Yeah, so usually we are saying if you're using our standard protocol and standard grinding conditions, the usual DNA fragment size will be approximately 30 kb or so, maybe even higher, but you can play around with that. That you can go higher if you're changing the grinding conditions a little bit. You definitely need to avoid grinding as much as possible, because we know that every grinding step is shearing, and shearing means degradation. So but it's possible when you're playing around. So we have customers who are doing this.
There was another question on grinding as well, about what is the ideal vessel to do the grinding in?
Oh, the one which doesn't break, I would say.
Okay.
Yeah, that is a very good question, because there are very cheap plates out there, right? I mean, I don't want to say anything wrong, but you're using ones from a certain company, which I can send you offline, because I don't want to make any commercials here. But it's really a very good question, because there are a lot of really cheap plates out there who are breaking all the time. But I can send you a link.
So we have recommendations that we could send?
Yeah, absolutely.
Okay, as a follow-on?
Exactly this problem.
All right, sounds great. When you're calculating the, when you're doing the calculations for sbeadex around the sample weight, is that dry sample weight?
Whatever you want. It doesn't matter, right? I mean, again, what is super important is we are starting with the standard DNA extraction at the beginning, and then we are looking what is a typical DNA yield, an average average DNA yield you're getting at the end. And if we are then seeing, okay, you don't need so much DNA, we can scale down the whole thing. And then it doesn't matter if it's fresh or not fresh. Or again, we are doing one test run at the beginning and having a look how it looks like, and then we are thinking about the next steps usually. And fresh leaves, I would rather think that the impact of the DNA fragment length is rather impacted by the fresh or not fresh leaf discussion, because fresh leaves are usually giving you higher DNA fragment length or longer fragments, because these fresh leaves are simply, again, you have more shearing with fresh leaves, unfortunately. That's what we found out in the past. So.
Okay. We have time probably for one more question here. And it's regarding the automated workflows, differences between the Hamilton and Kingfisher. There was a difference in in timing between those two workflows, and there was a question around what causes that difference in time between the Hamilton and the Kingfisher.
Yeah, it's a technology again. A typical Hamilton instrument is a liquid handler, and the liquid handler takes a lot of time to move the head or around, you know, because you have to go to the buffers, take the tips up, put them down. Then you have just an 8-channel head maybe, or 1-channel head. Can be pretty slow. You know, and but a magnetic particle handler like the Kingfisher, where you have a carousel in the middle and you're just moving the beads from one plate to the next one, this can be pretty quick. For example, for one project, we have developed a 50-minute protocol on a Kingfisher, right, which has been providing sufficient DNA purities for qPCR. So it's simply the technology, and the Kingfisher is very often used in this ag bio world, and simply it's much quicker. And by the way, the other big benefit of the Kingfisher is that you do not have any risk of blocked tips, right? Because you do not use any tips, you're not moving the liquids, you're moving just the particles from plate to plate, and therefore you're simply not running into any issues with blocked tips and a big mess on your robot because of that. Because that's a really big problem for every DNA extraction. That liquid handlers are sometimes tricky with these block tips.
All right. Well, thank you. There are definitely some questions here from our audience that we did not have time to get to, but we will be following up with you via email to answer those questions. We will also be sending out a link to the recorded webinar and to a downloadable copy of the slide deck for you. So please keep an eye on your inbox, audience, for that coming to you within the next couple of days. I'd like to thank our presenters for today, Dietrich Köster and Rob Brazos. We appreciate your time. Thank you to the audience for joining us. We appreciate it. Thank you for the great questions and for your engagement. And we'll go ahead and close the event now.
Thank you very much. Best regards. Stay safe, everybody. Thank you.
Bye, everybody. Take care.